I'm Nazarian from DC Children's. It is a pleasure to work with the DRC, and one of the first ideas of launching a project called Open DIPG. DIPG, diffuse intrinsic Pontine glioma, as Adams and my other colleagues mentioned, it accounts for about 15% of all pediatric CNS tumors. It is a pediatric disease. The peak age of onset is between the ages of 5 to 10. Over 95% of children who are diagnosed with DIPG die within 1 year of diagnosis. Gabriella Miller, who was diagnosed with DIPG and lost her life to DIPG, has been an inspiration behind the Data Resource Center and the Gabriella Miller's Kids First Act that we are here to talk about today. There has been a recent publication that discussed 1,000 genomes that discusses pediatric high-grade gliomas and low-grade gliomas. On the right, you see the surface plot that describes some of the mutations that are associated with pediatric brain tumors. As you can see, the majority of these mutations are associated with histone genes, highlighted here in dark and light green, and also blue, dark blue in H3G34. What is important to note here is that a good portion of these patients are wild type, or they have mutations that we have not yet recognized. The 1,000 genome that was published last year was a huge effort and huge step forward for understanding and for cataloging pediatric brain tumor genome. If you look at the history and why it's been taking us so long to develop 1,000 genome, which is really not much when you compare these to adult diseases, is that the complexity of the biology, but also the lack of the access to the data resource centers and specimens. Because of the work that has been done, we now have zip codes that are associated with mutations of these childhood brain tumors. In dark green here in the pons or the brain stem, you see mutations that are associated with H3K27M. This is a mutation of histone that is typically gives rise to Pontine tumors or DIPGs, a tumor that Gabriella Miller had. The lighter blue colors here are mutations with H3G34. These are mutations that are typically manifest in the cortex, and it shows that biology actually translates into the different anatomical locations of the brain. Again, these are data that have been generated within the past 5 years. We typically classify these tumors as pediatric brain gliomas 5 years, and now we have a mutation that's associated with them. And these mutations actually make sense. If you look at the subcategory of these mutations, with G34RV, which I mentioned that are cortical tumors, these are typically more older children, more younger adults. If you look at H31 mutations, these are typically younger children. And we believe that data has not been completely looked into, and if we want to look into them, we should be able to find more subtypes. More importantly, they actually affect the outcome. The children who have histone wild type, they tend to live longer, and the children who have histone 3.3 and 3.1 mutation have the worst outcome. Again, we're going from 10 years ago when these patients were radiographically diagnosed, had a brain tumor, a brain glioma, as was diagnosed by a pathologist. Now we have genomic mutations. We can actually predict the outcome. But we haven't done enough work. And the reason is that, again, if you look at the timeline of the DIPG, if you look at the Google search for the word and how often did it show up in the past 15 years, the word DIPG, you can see that in 2006, there wasn't really much mention of DIPG. And this is a Google search that is looking at all of the published and available material that has mentioned the word DIPG. It was really until 2011, 2009 that the word DIPG started showing up. And if you look at why it was, is that the first models, the first rat models, were called brainstem gliomas. Even the word DIPG was not used very often then. The first brainstem glioma rat models came about in 2011. They were followed by cortical xenograft models. This is when we started injecting the tumor from the patient into the brain of the mouse to make a model that we could study that would be more representative of the patient. Then very recently, we started developing DIPG xenografts. Now, these were not brainstem gliomas or cortical tumors, but specifically Pontine tumors. That was followed by cell lines, DIPG cell lines, that now we could take 1,000 cell lines, 1,000 cells in a petri dish and test them with 500 different drugs. Something that we cannot do with a pediatric rare disease, such that it would highly limit the clinical trials that we intend to do. So now we have started to develop models, preclinical models that we can test. As a result, the spike in the word DIPG with the Google search. The red line here shows the publications. These are scientific publications that are meaningful biologically. Up to 2009, there have been hundreds of papers, clinical papers that have been published showing that every clinical trial has failed for DIPGs. The red line shows here only scientific papers that have started looking at the biology of the tumor. To 2009, there were really 8 scientific papers that were discussing DIPG, but we get 8 scientific papers on DIPGs now every 2 or 3 months. A huge success for a rare disease that we really had nothing, we knew nothing about before. And I have divided this into 3 phases. The early animal models, the robust preclinical models, again, the cell lines that have been developed from patients, the cell lines that truly represent the mutations that patients with DIPG carry, and we can test an availability of multiple cell lines as far as histone mutation patterns or P53 mutation or ACVR mutation that my colleague mentioned. But the latest phase is the clinical translation. The question we have is that given the fact that we had successful past 10 years to develop models and data for the DIPG, why have we not made a dent in the clinical translation? Why we have not been successful? And the question that I have and my colleagues have is that what is that is truly delaying the clinical translation if we have so much knowledge that we have acquired in the past few years? One of the major components of lack of acceleration in discovery and translational application of the data is the siloed data. We have genomic data, RNA data, epigenomic, which tends to drive most of the pediatric brain tumors, the clinical data, all siloed in different institutions and in different hardware, different computers. There's one silo that I'm showing here for genome, which is really not true. For DIPGs and for many other pediatric diseases, there are hundreds of silos that are holding and storing the genomic data. Without the access and without the connection between this data, we will make no change in the current status of patients who are diagnosed with DIPG. At this, you can see in the image that the farmer has connected silos, and this is something that we really have not done to date. And the idea of the Open DIPG has been to connect them and to make sense. Specifically, if you look at the genome data and the clinical data, how different are they? If you only look at the genomic data, we will never understand the etiology of the disease. Here is a good example. This is one species in 2 developmental stages. They have identical genome. We could sequence the genome of the caterpillar and the butterfly for the next 200 years. They would be exactly identical. What is different about them is the epigenome, is how those genes have been reprogrammed during development and have given rise to new proteins that is making a different morphology. The genome is absolutely the same, the epigenome and the proteome is what is changed. So that's brings the question of, are we looking at different epigenome? Are we looking at different proteome of these diseases? Radiographically, this is a black and white image showing the Pontine tumor in the patient on the left. On the right, you see the extension of the tumor, the ventricle of the patient. But this is purely radiographic, black and white, as simple as information that we can get in 2018. But we think that our children deserve better. I think we have the resources and we just have to connect them. And this is the idea of the project Open DIPG. The word that you will hear today very often is data harmonization. And harmonization means really connecting the data types that make a subset of data, for example, different platforms that have been used to create the genomic data, or the genome, or the type of data, the genome, RNA, epigenome, clinical, and proteome, and many, many, many other data that is rising. As a result of Gabriella Miller's first data resources, we can actually imagine bigger and bigger scenarios and stories. We can actually think big and throw in as many as omes that we want, genome, proteome, secretome, models, preclinical models, and add them to this, because the idea is that we can combine them and we should be able to harmonize this data. And at the end, what you will get is a simple page that, to me as a researcher, will tell me what is the pathway that is driving this particular tumor. And I cannot do this in the laboratory unless I have the data connected and unless I have those data harmonized. It would make sense to me as a user that I can see a simple page that tells me a simple message of which pathway is activated. And unless we cannot do that in the laboratory, that cannot get translated into the clinical setting. Connection, translation, and harmonization is very important if we are truly want to make a dent in the current status of pediatric brain tumors. And that has led us to start and to initiate the project Open DIPG initiative. This is a project that we spoke about last year, and we are very proud to show that we've had a good progress in amassing the data that we suggested before. The idea for the Open DIPG is that we start from one challenging disease. We will tackle that by truly gathering the data under one roof, have them harmonized, and have them accessible by researchers, and make that open initiative as a model for other rare diseases, for other diseases, even adult diseases, can use the same model to tackle the particular diseases. The data will be shared in real time and available. The large goals of Open DIPG are to gather all of the published and newly generated data. You may think that published data are accessible. That is an NIH mandate, specifically if the research was funded by the NIH. They are accessible, but they're fragmented. Different institutions and different PIs, myself, we publish data, genome data, proteome data, and we deposit those on NIH-mandated websites. The same thing is done by our colleagues in Europe or in Canada. However, they're not connected. We want those to be connected. The newly generated data should be connected and should be added to this genome and the information. We want to harmonize them and integrate them. We want to make data available and usable. Availability is as important as usability. Data have to be useful. Harmonization, which we mentioned, will make those data useful. Data should be linked to the clinical data. You should be able, an end user should go to a computer and ask a simple question. How many 3-year-old African American boys have histone 3 mutation. We currently cannot do this. We can look at histone mutation, but we cannot ask clinical information. How many of these children received albinostat for the mutation? We do not have that connectivity. The idea is to make that connection across all of the clinical data, across the phenome, which would help us to understand the genome. But more importantly, again, because the DRC has created such a fantastic model, we can dream bigger. The idea is that once we find the pathways, we should be able to take those hypotheses and test them in the lab. We should be able to take cell lines, mouse models of DIPG, characterize them as a genome, epigenome, and proteome, add those data to the DRC, and link and harmonize them, such that an end user would say, how many 3-year-olds with H31 mutations received albinostat, and what was the outcome? Requests for the cell lines that have been characterized, that have the same mutations, that can be tested in the laboratory. In addition to albinostat, different drug can be used to accelerate the translation of the findings that we have, to make the data more alive than a set of data that will be sitting in a cold hard drive. And we've been very successful. The Open DIPG has been successful to receive and harmonize the data that from institutions that are shown on the right. We're working with the institution on the left. They've been very kind with sharing the data with us. You see the Pacific Pediatric Oncology Consortium, the PNOC. These are, again, recently generated data, unpublished data that are being used and accumulated under the same roof as we suggested and dreamed for to be the mission of the Open DIPG. The data that we have collected, again, under one roof, under one data resource center, represents over 800 DIPG samples. This is the largest DIPG genome and molecular data that has been collected under one roof. And as I mentioned, many national and international institutions have been contributing to this. They've been contributing to this because the resources have been generated and collected by the NIH and supported by the NIH and by our colleagues here at DRC. These are the type of data that currently exist, and again, it emphasizes the need for harmonization of the data. The platforms that this data was run for are listed here: Affymetrix, CHIPS, Agilent, Illumina, for those of you who are familiar with the platforms. And these data are typically, for me as a scientist, as a researcher, if I take two sets of data that are run on two different platforms, it will be really difficult for me to connect those two data sets together to make sense. But the idea is for the DRC, for the Open DIPG, to harmonize the data amongst 800 or so patient samples, subjects that I mentioned that currently exist at the Open DIPG platform. These data include genome, exome, exome plus genome. Again, correlation between the genome and exome would be really helpful for clinical trials where the question is, should the biopsy be subjected to exome or genome, where one is much faster or much cheaper, but are we getting more genomic information, more mutation information from one or another? So it's helping to drive the future of clinical trials. Harmonization and looking at the mutation of the samples that we have now is shown here on the left. This depicts the histone mutation, the H33 mutation patients, the H31, and the patients that we have not categorized under the H3. But perhaps the most important aspect of data that have been collected is here: pre-treated samples. This is very important and very informative for the current clinical trials, current and future clinical trials. One of the major components, one of the major questions that we've been having in clinical trials is that how much of the harm that is done to the patient and how much of the genomic changes that we see in the tumor is caused by the treatment that the patient receives. These preclinical samples, pre-treatment samples, treatment-naive samples that have been characterized and will be linked to the post-treatment samples will give us a good idea of how much of the tumor evolution is a true evolution of the tumor and how much of it is done because of our intervention that could potentially be prevented. Again, the data has come from many of our collaborators, including the PNOC that has active trials on DIPGs. We have the grading for the patients, the tumor grades, 2, 3, and 4. We also have we have organized them based on the location, brainstem versus the midline glioma, including some of the clinical data with the patient. Histone mutations again, and again confirming that a good portion of these patients are histone wild type, meaning the biology of these patients, and we have so much more work to do. We have amassed the copy number variation of these tumors, and perhaps some of the patients who have more of the uneventful genome, they may have copy number variation that can be detected. We're looking at mutual exclusivity, which simply means that in how many of the patients, when we find a particular mutation, the second mutation can or cannot be present with the same mutation. Meaning that, for example, if you have a histone 3 mutation, how many of those patients would potentially have a p53 mutation? This is very important for precision medicine. This is very important when we collect very small biopsies from these children and we subject that biopsy to genome analysis. This would help the clinician to say, we do a quick first analysis. If this patient has H3 mutation, we should absolutely look for p53 mutation because they always show up together. Or we should not look for ACVR mutation because they are mutually exclusive. We also look at partner mutations. Partner mutations are very important for clinical trials, again, where we want to have a rapid analysis of the samples, rapid identification of the mutations associated with the patients, and also looking to see which other mutations should we expect to see in these patients. And this can be done only as a result that we actually have a very large number of DIPG and pediatric high-grade samples. A good example of partner mutations would be a very non-invasive method of liquid biopsy, which again has been developed through a collaboration with CVTTC, PNOC, and Children's and BloodPack, which lead into the data that has been generated through the DRC. The idea of liquid biopsy is that you can use blood, urine, and CSF, the liquid biome, to look at a particular mutation, in this case a histone. Sample blood test, a 1 ml blood test is taken from a patient, or half an ml of CSF is analyzed on the dPCR, and the mutations that have been shed by the tumor cells in the brain will be collected and analyzed. We have shown that we can actually detect all of these mutations which are associated with DIPGs. The importance of this is that in future, a simple blood test can tell the clinician that the patient has histone 33 mutation. And from the data that is coming out of DRC and Open DIPG, we can predict which partner mutation could be there and which could not be there through the data that I showed you through mutual exclusivity and partner mutations. And patient can be put on a specific clinical trial, meaning that that could be the future of the precision medicine and personalized therapy. Again, one of the major aspects of this is to include unpublished and newly published and upcoming data. This is the genome profiling data from PNOC that shows a circle plot that shows the genome aberration of the biopsy that was taken from a patient at diagnosis. This patient had not received any treatment when this biopsy was taken and when we looked at the mutations of this patient. The biopsy was taken at progression. You see that the genomic change, the genomic pattern and changes are very diverse and they have changed from the primary tumor. We've also developed cell line on the same patient. You can see genomic changes and genomic aberration between the cell line, between the primary tumor, and between the progression. Unfortunately, this patient died from DIPG, from the disease, and we looked at the tumor post-mortem. This is a post-mortem analysis of the tumor from different sites within the brain, from different metastatic sites within the brain. As you can appreciate here, the tumor's genome and the genome mutation landscape is changing. This was not known before, or if it was, we didn't actually have a good genome map of a patient upfront, at progression, post-treatment, and also at autopsy. The cell line that we have been created here has been analyzed and data has been linked and stored at the DRC. It allows us to help this tumor evolution from upfront, from primary to postmortem. It will help us to understand why did the tumor change and how did it change. The analysis of all of these tumors will help us to understand which ones are really important changes that we are seeing here and which ones are the driver mutations. Many of these changes may be important, many of them may not. But without connectivity of this data, connecting them to the preclinical model, which is the cell lines in this case, you would never be able to understand. As I mentioned, the DRC and the Gabriella Miller CRISPR has really allowed us to think big. Just collecting the data, harmonizing them, having them accessible will not help the clinic, as I mentioned. There's a huge effort underway by our colleagues at CBTC and DC Children's to make this more translatable by taking the samples from the patients, making the mouse models, look at the survival, put the mouse models under the same treatments that the patients have, to characterize those mice, characterize their tumor, and digitize and connect all of this histological data with the genomic data that has been created. Collectively, these efforts will accelerate discovery. We highly believe that the connection and accessibility will be important. The vision of Open DIPG will serve as a platform to establish the mission of the Gabriella Miller CRISPR Pediatric Data Resource Center. It will be a good example for many other diseases that are subjects of studies today. We believe that data are powerful when they are shared, harmonized, and accessible. As Adam mentioned, the thousands of the books in the library cannot be powerful unless they're connected and they're used. Open DIPG will serve as a centralized database to assemble specified data sources together into one location. It will provide easy access for query with researchers without bioinformatics support. Most of us, as clinicians, physicians, researchers, we do not have the bioinformatics support. We do not have the bioinformatic understanding. All we want to do is to look at the genomic profiles and the pathways that they drive and hypothesize a drug that would work. Open DIPG mission is to accelerate discovery by consolidating knowledge, and that's the basis of the Open DIPG. With that, I would like to thank patients and families that have contributed both financially and by contributing the tumor samples that we've been able to use, and many of our colleagues and foundations.